This meeting is now being recorded.
Well, good evening. Welcome to the EDS Awareness Educational Series webinar. My name is John Furman. Before we introduce our speaker this evening, Dan Doherty, we'd like to give an overview of our free program to those attending for the first time. So we'll go over who we are, what is our program, some upcoming events, introduce our speaker for this evening, and then have a Q&A. So again, my name is John Furman. I manage the National EDS Awareness Program based in Cincinnati, Ohio. My daughter Deanna, who is, unfortunately isn't on the call this evening, leads the Cleveland, Ohio EDS support group. She was diagnosed with EDS in 2008, the same year my wife Carol passed away with breast cancer. I was a caregiver for my wife who struggled with undiagnosed EDS for over 30 years. We introduced our program at the 2012 EDNF conference to help EDSers form independent local support groups and spread EDS awareness in their communities. We've started over 100 groups to date. Each group is given their own free website with a link from the directory and map. We received feedback that conferences provide valuable information and social opportunities that many cannot afford physically or financially to attend in person. So we decided to bring the speakers to you through the EDS Awareness Educational Series. We meet the first and third Tuesday, typically at 7:30 Eastern Standard Time. All the programs are free. The meeting announcements and whenever possible, the webinar recordings will be posted on our site at edsawareness.com for later replay. You can receive email announcements for future sessions by requesting the free guide on our site. Tonight's presentation is sponsored by Body Support Store, where you'll find over 250 products selected by EDSers for EDSers. The store is the only funding for the program and usually covers our monthly web fees. Please visit our store and check out the helpful products that we offer. We also have a webinar sponsor. Elizabeth Shaw actually did a Elder Danlos Hoofbeat Run and Walk in August at the King Ferry, New York, and she raised some money to help fund and support our programs. So we thank her for that. So just a general disclaimer. This presentation contains general information about EDS. Members of the EDS community voluntarily participate in this program. The information is not advice. If you're having medical problems now, please call 911 or the emergency services in your location. Always consult your doctor before making any changes to your treatment. We'd like for you to mark your calendars for October 18th. We're looking forward to Dr. Collins. She's both a physician and an EDS patient. She is very involved in the EDS community as a speaker and advocate. To view all of our upcoming speakers, see the webinar page at edsawareness.com for full schedule. For those attending live this evening, there will be an opportunity for Q&A after the presentation. Add your question at any time during the presentation by clicking on the Q&A icon at the top of your screen. After typing the question, click the orange button to submit. Our speaker this evening is Dan Doherty from Alleles Corporation. He's going to be talking about pharmacogenetics testing to assess altered drug metabolism, and that's a mouthful. Actually, Dan has been with Genetics since 2006, working with patients and clinicians to improve medication outcomes with U-Script, an online clinical decision support tool. Dan has been supportive of the EDS community as a sponsor at the EDS conferences for several years. In fact, Dan has actually been our neighbor at the last 5 conferences. So we're really looking forward to hear Dan talk about this program. So Dan, I'm going to bring up your slides, and I'm going to turn over the microphone to you.
Great. Thank you, John. Can you hear me okay?
Yes, we can hear you fine.
Okay, so we'll go ahead and jump in here. I'd like to begin with key terms because there are a lot of terms that may be new to you or not, depending on your level of understanding with pharmacogenetics. So to begin, probably the biggest and toughest term is just pharmacogenetics itself. And what that means is it's really a combination of 3 words: pharma, meaning medications, co, meaning with, and then genetics. So it's just a big word for medications in combination with genetics. And it's the branch of pharmacology concerned with the effect of genetic factors on reactions to drugs. The next term you'll hear often from me during this presentation is polypharmacy. And again, that's a dual term. It means poly, meaning more than one, and pharmacy, meaning exactly like it sounds, like drugs, pharmacy. And it really just means multiple medications. So polypharmacy, multiple medications. A polypharmacy patient is a patient taking multiple medications. So a lot of Ehlers-Danlos patients, for example, could be described as polypharmacy patients. The majority probably would be described as polypharmacy patients. ADE is just short for adverse drug event. That is when an individual takes a drug and something occurs that is not anticipated with that drug. Efficacy, this is a term you're probably very well familiar with. It's the ability to produce a desired or intended result in a drug response. And then we're going to get a little bit more technical now. Enzymes are, these are something we'll be discussing a lot. In this part you don't have to remember, but enzymes are just molecules or proteins that accelerate or catalyze chemical reactions. So in what we'll be talking about today, these enzymes are needed to break down drugs in the body so that they can leave the body, so the drugs don't remain in the system longer than intended. Allele, now we're getting very technical. You don't have to memorize this, but just I'll be discussing this, and you'll want to kind of keep this in mind as we go. So an allele is 1 or 2 more alternative forms of a gene that arise by mutation and are found in the same place on a chromosome. So basically, your alleles determine your genotype, and allele means, I think the Greek term is other, the Latin term allele stands for other. And then we have phenotype, which means the origins are to show type, and again, it's a compound word, pheno and type. Pheno means to show, and then type is type. And for our purposes, it's going to be, the phenotype will refer to the lab test results. And then another large term here is cytochrome enzymes, and these are shortened for CYP450, or also CYP450, or just CYPs. It sounds like SIP, but it's CYP. And these are essential for the breakdown of by far the majority of drugs in the body. These are the enzymes we'll be discussing today. Lastly, on key terms is CYP2D6. So that, when I say CYP, that means cytochrome P450, or CYP2D6. And it can be shortened for just 2D6 all by itself. It's the name of one of the most significant of the cytochrome P450 enzymes. It metabolizes about 25% of medications, some more than others, but to some degree about 25% of all meds on the market. And it has a highly significant amount of genetic variation between people, and that's what makes it one of the most important, if not the most important, cytochrome P450 enzymes. So it's the one you'll hear me discuss the most, but there are many of these enzymes. So we'll go over kind of a couple things here before we get started. So learning objectives. We'll review how drugs work in the body relative to adverse drug reactions and lack of efficacy, how your genetics play a role in drug metabolism. The specific focus will be on pharmacogenetics, in medications plus genetics, in regards to the cytochrome P450 enzymes. We'll look at drug-drug interactions at the enzyme level, which can have to do with inhibitors and inducers, and we'll get into that. And then more generally, when to consider pharmacogenetic testing for yourself or a family member, how to approach your physician about getting tested, and then finally, how to approach test results using existing online materials that are free on the web or an online clinical decision support tool. And that's made by our company, or my company, Genelex. Speaking of which, conflicts of interest, definitely. So I work for a lab that provides pharmacogenetic testing. So I certainly have a slant towards our lab because I feel like we do very good work, and I'm very proud to work for this company. So certainly in regards to other companies, I'm going to have that slant. So keep that in mind as I give this discussion. I work for this particular lab that does this testing. So Genelex also developed UScript, which is a cumulative drug-gene interaction software, and we call it a clinical decision support tool. And that is specific to our lab. There's no other lab that makes a cumulative drug-gene interaction software, and that will come up towards the end of the talk. And again, it'll sound a little bit like a commercial because we're the only lab that makes it. So again, keep that in mind that I'm going to have kind of that slant, but most of the discussion will not be that way. It'll be strictly educational up until that point. Probably the most important thing to keep in mind, I'm not a medical professional. My experience over the last decade has come from discussing pharmacogenetics with lab personnel here at Genelex, with the pharmacists that work with us, physicians, and patients. So consult your physician when considering pharmacogenetic testing. Most importantly, do not make any changes to your medications based on this presentation. Consult your physician. So there may be times where you hear me talk about a medication that you might be taking that could interfere with specific other medications. It doesn't mean stop taking that medication. Your doctor may already be aware of that potential for interaction and may be changing or have already changed your dose accordingly. So again, absolutely don't make any changes whatsoever. Talk to your doctor first, because odds are they have a very good understanding of what's going on already that you may not be aware of. And then some acknowledgements here. First, the Coalition Against Pediatric Pain, or TCAP. They introduced us initially to the Ehlers-Danlos community. The Chiari and Syringomyelia Foundation has been great too. TCAP and CSF have both allowed our lab to present to physicians who are involved with Ehlers-Danlos, hypermobile, Chiari groups, as well as POTS and Mast-Cell. It's really been helpful to us because we had very little understanding about these conditions, and has given us an opportunity to talk to patients who have a lot of polypharmacy, who are taking multiple medications, to understand the importance of potential importance of pharmacogenetics for some of those individuals. And then the Ehlers-Danlos Society, certainly, or EDNF, I guess, in the past. And like John said, we've been presenting there for quite a few years now, right next door to John, so you can always find us there. And finally, to John and Deanna at EDS Awareness, who have been really great in allowing us to speak to you this evening. So presentation outline for the evening. We'll do a quick crash course, just kind of a quick 15-minute review for those of you who are sitting through an entire presentation, which may be about an hour plus, could potentially be uncomfortable. So I'll kind of do something very quick to just give you a quick overview in case you can't stay with us for the whole thing. But you can always catch the recorded video later on John's website at edsawareness.com. So we'll review what the cytochrome P450 enzymes are. How did pharmacogenetic testing come about initially? The body is a constant cycle of experiment. Polypharmacy and pharmacogenetics in the Ehlers-Danlos community, specifically the pharmacogenetic landscape in approaching and ordering testing, a UScript clinical decision support tool that we discussed a little bit earlier, and then final thoughts and then questions at the end. A quick thank you for getting through the key terms with me and all that. So a quick cartoon here as we officially jump in. So here we go into our crash course. So our best effort to impact drug response, and this is us as in all of us. It's simply, compliance, taking the drug, taking it according to meal times, which can be very important, and according to sleep. As many of you may already know who are polypharmacy patients, if you take drugs outside of meal times or certain drugs at the wrong time of day, it can really impact how you respond to a drug. So you definitely want to keep in communication with your pharmacist to ensure that you're adhering to that, because that can play a major role in how you respond to medications. And then drug-drug interactions, inhibition and induction. These are things we'll discuss a little bit later that can have a major impact on how you respond to drugs if you're taking multiple meds, potentially. And then finally, pharmacogenetics, and that's what most of the talk will be about today, that your genetics impact or can impact how you respond to medications. Again, pharmacogenetics just means medications plus genes, and these are specifically going to be drug metabolism or metabolizing enzymes that we'll discuss. And the key, really, two things you want to walk away with today is that people do not respond to medication the same way. And you probably already know that as an Ehlers-Danlos patient. You hear that on the chat rooms and billboards that what seems to work for one patient doesn't necessarily work for another patient. And we'll be discussing a little bit of why that is, at least in regards to pharmacogenetics. And then the second one is, again, we inherit our response to drugs, that genetics play a role in how we respond to medications. And this is a pretty good visual of what that looks like. You could have a patient group, so by patients, just really anybody, right? Anybody taking medications. And these people could have the same diagnosis and be taking the same prescription, but for some of the people, the drug could be toxic but still beneficial. For some, the drug could be toxic but not beneficial. For some, the drug could not be toxic and be beneficial. And for some, the drug could not be toxic and not be beneficial. So, again, you can have the same group of folks, but depending on many variables, we can have different responses to drugs. And genetics play a role with some of that, not all of that, but with some of that. So genetics and pharmaceuticals, 75% of all medications are processed by genetic-specific enzymes, the degree of which varies depending on the medication. And importantly, 90+% of the population have genetic variations that increase their risk for adverse drug events. So in other words, this is a very typical issue. Over 90% of the population has these enzymes functioning differently than normal. The degree of which is what's key, and then if they happen to be taking a medication that is dependent on an enzyme, they have functioning differently than normal. So this is a very common issue, and that's not just specific to patients with Ehlers-Danlos syndrome or other conditions. We have a study results how common are drug and gene interactions, and the study showed that drug-gene interactions, where there's a genetic component to a response to a drug that was negative, accounted for about 33% of adverse drug events. So this is very significant. So about a third of the time when someone is experiencing an adverse drug event, that there's a genetic component to that. And the FDA is keenly aware of this. They've put black box warnings on certain drugs suggesting genetic testing for pharmacogenetic testing. They've done it for warfarin, for codeine, for Plavix, so a number of different drugs. And I think they have warnings now on about 120 different medications, some black box, some not black box warnings. And the FDA has acknowledged that these interactions, just adverse drug events, are costing us somewhere between $30 billion and $136 billion per year. And we know that adverse drug events due to genetics play a role in about a third of that. So these are very serious interactions that are costing the U.S. healthcare system a great deal of money. The FDA is aware of it, and it's affecting many people we know day to day in our lives who are taking multiple meds. As far as which drugs are dependent on these enzymes, there are hundreds and hundreds of drugs dependent on these enzymes. About approximately 75% of all drugs on the market, plus depending on which enzymes. And I'm going to go into which some of these drugs are, but these cover cardiology drugs, psychiatry drugs, pain, some cough medicines, PPIs for GERD, antihistamines. So, this will cover meds that are prescribed for mast cell, for EDS, for tachycardia, for POTS. It's a little bit of the kitchen sink, all sorts of different medications. The main enzymes we're going to discuss today are the cytochrome P450 enzymes, and there's really 5 that have the greatest amount of genetic variability from one person to another, meaning you could have a next-door neighbor who's functioning normal for this enzyme, but the person who lives next to them is not. And these are really the 5 most significant enzymes in that they have the greatest amount of genetic variability between people, but also they just statistically cover the most medications. Medications are more dependent on these enzymes than any others for metabolization. So we have 2D6, 2C19, 2C9, 3A4, 3A5. And 2D6 is the one I'll be talking about the most just because it covers so many medications, about 25%, and has the greatest amount of genetic variability that's clinically significant. We see from our own data and our internal data that about 93% of tested patients who have tested with our lab have at least one of the 5 enzymes functioning differently than normal. And interestingly, it's not just patients who've tested with our lab who have had adverse drug reactions, it's our own staff. So our own staff here at the lab, we see that same number. So we really believe that 93% is not just people who have tested with us who've had adverse drug reactions, but we're actually seeing that in our lab staff who haven't necessarily had adverse drug reactions. And interestingly, the majority of all these, about 41% of the population, will have typically 2 enzymes functioning differently than normal. So of those 5, about 41% of just what we believe to be the general population has 2 of these enzymes functioning differently than normal. So this is a very, very common thing in the general population. And now we'll look at the effect of drug metabolism on excretion. And I'm going to add my cursor here so you can see that. So this might look a little daunting, but bear with me here. This is just a picture of the liver. The majority of the cytochrome P450 enzymes are made by the liver, but they can actually be made throughout the body, but by far the majority are made in the liver. And basically this picture is a picture of the drug themselves. The drug is in blue, and as on this side, where my cursor is on this side, if the drug is able to be metabolized by the enzymes, it's then able to leave the body. And this lower picture here is excretion. It's basically urination. So that's what we're looking at here. On the other side, we see the drug where the enzymes are not being made by the body, and the blue drug here goes down and wants to leave the body through excretion, but it can't because it hasn't been metabolized by the body. It can't actually leave the body through urination, and that creates a problem where there's a potential for drug buildup. And this is what that looks like. So I'm going to draw a line here. And what we call normal metabolizer, this is what we would expect, the green here, that the drug would be metabolized by these enzymes and be able to be urinated from the body. That's what we want. But for some individuals where they're not making that enzyme and they take an active drug, the drug cannot leave the body. And so there's a potential for drug accumulation or even toxicity if the drug continues to build up because it can't leave the body. And this is really the gist of what we're talking about. This is what's going on in the body. This is where our concerns lie. We really want to identify the individuals who are not making these enzymes, so we make sure that they're not taking drugs where this is what's happening. We want to make sure that they're able to metabolize drugs. So now we'll talk about the phenotypes. And again, the phenotype is just simplified for our purposes. It's the test result. And the phenotypes are a term for a genetic difference between individuals. So we have these phenotypes have been developed by a group called CPIC, and they kind of are a group that comes together to make clinical decisions regarding genetics, and they've kind of deemed that these are going to be the four main names for phenotypes or test results. So one is normal metabolizer, or NM. This is the norm. This is what typically the majority of the population would be what we call, quote, a normal metabolizer. Then we have intermediate metabolizer. This person makes about half the typical amount of the enzyme. And I'm going to get into these in more detail in a moment. And then poor metabolizer, these folks make little to none of the enzyme. These are the folks we worry the most about. And then finally, ultra-rapid metabolizers. They make much more of the enzyme than normal. Depending on whether they're taking a prodrug or an active drug, we have concern for these individuals too. So these two are the ones we worry the most about, where they can really have the most potential for adverse drug events. And so now we're going to start getting a little bit technical here, but bear with me, it's not as technical as it looks. So if we go by what I said here earlier with PM, or poor metabolizer, so this is basically a graph that shows plasma levels for nortriptyline, which is an antidepressant, and we have 0 to 60. So these are the plasma levels going from 0 to high, and then we have the hours in which the individual experienced the result of their genetics in relation to nortriptyline, okay? So our poor metabolizers, or actually let's start with our more normal metabolizers here in orange. We see that their level is this is kind of the level expected. This is what we want to see when a patient takes a standard dose of a drug as made by the pharmaceutical company and as agreed upon, dosage by the FDA. So this is what, your doctor wants to see happen. We're going to see the drug kind of spike and then slowly go down as it is metabolized and leaves the body. Our intermediate metabolizers, we see this yellow bar, and it's higher. They have a higher amount of that drug. And then we have the poor metabolizers, which are the highest. That drug is going to spike because it's not being metabolized, and it's going to stay in the body at a much higher level over many, many more hours. In fact, days here, right? 72 hours is 3 days. And then finally, our ultra-rapid metabolizers, they get very little relief from the drug, and it leaves the body right away. And I'm going to show kind of why this is happening here for each of the phenotypes. So here's our normal metabolizer, right? They have 2 functioning alleles passed down from mom and dad, and we say that these individuals can take medications at recommended FDA dosage as made by the pharmaceutical company. So genetically we say, great, they should have no problem with the drug as made by the pharmaceutical company, but they can still have allergies or side effects with the medication. Certainly for those who have mast cell or other allergies, you can certainly still have an allergic reaction to drugs, which the cytochrome P450 enzymes will not play a role in. So keep in mind, allergies are separate. Allergies, depending on the allergy, can come and go during your lifetime, whereas with these enzymes, you, for the most part, are born with these enzymes, and they remain that way literally till the day you die. These are genetics, so that they're not going to, your baseline genetic capability for making these enzymes will really not vary. And I kind of have a picture to give you an idea of what this looks like. In the drug level, I'm kind of calling or making the color of the shirt and the level of the shirt to give you an idea. And hopefully that's helpful and not confusing. We'll see. So again, our normal metabolizer here is here in orange, and we see, we expect that drug level is going to hit around, maybe 35, and then it's going to go down as we would hope. Our next phenotype here is our intermediate metabolizer, and we see the drug level go up here for an active drug. These individuals have one functioning allele and one non-functioning allele, and again, one is passed down from mother and one from father. These individuals make less enzyme than what we call, quote, the normal metabolizers, so they make about half the typical amount. For active drugs, which are most drugs on the market, but not all, and we'll talk a little bit about that later, the drug level will typically increase because they're not metabolizing the drug, so it's not, some of the drug is leaving the body, but not at quite the rate we would expect. So they're going to have more of the drug available in the body than anticipated. And so for these individuals, depending on the drug, often a lower dose may be appropriate and more suitable to them for the right efficacy. For prodrugs, the active metabolite will potentially decrease, so a higher dose may be appropriate. A prodrug is basically a drug that is a two-stage drug. It has an active metabolite. So kind of think of it as a drug where there's an outer shell and an inner shell. For active drugs, they begin working immediately in the body. For prodrugs, the outside shell needs to sort of be metabolized by the body first. So the inside of that, the inside shell, I guess you could say, can be released into the body. And so these two drugs, type of drugs, active drugs versus prodrugs, can work very differently in the body. And it's important if you're taking multiple meds, especially pain meds, a lot of opiates or prodrugs, to begin to understand that if you're not already familiar with it. And so here's our intermediate metabolizer, this yellow line. When they take nortriptyline, and they're an intermediate metabolizer for the 2D6 enzyme that nortriptyline is dependent on, they're going to have a higher level of this drug in the body. They're not likely to be toxic, but they're going to have drug accumulation and may or may not feel the effects of that. So they may be perfectly therapeutic at that level, depending on the individual. Height, weight could play a role with it, age as well. So just being an intermediate metabolizer doesn't mean you're necessarily going to be toxic. That's not likely the case. But there's potential for a higher amount of the drug to be present in the body for more drug accumulation. And our poor metabolizers, and this is kind of an extreme picture here, but this would show, someone who has no functioning alleles. These enzymes make little to no, or these individuals make little to none of the enzyme. And so for active drugs, there's risks of toxicity. Depending on the drug, if the drug is highly dependent on an enzyme that the individual doesn't make, the drug level can increase significantly. So a lower dose, or probably even better, really, an alternate drug may be the most appropriate choice. And then for prodrugs, it's kind of really the opposite. There's a potential lack of efficacy, where the active metabolite, the part of the drug that's kind of inside that outer shell, may not reach the bloodstream at all, or very little, so an alternate drug would be more appropriate. And here's our poor metabolizers, the gray line here. We're going to see that the drug level really spikes. So, this person is getting almost like double the dose of nortriptyline than expected, and then it stays in the body for much, much longer. Over here we see the drug level is very low after 3 days, but for this individual it's a much higher rate. So this individual, if they're a poor metabolizer for 2D6 and they take nortriptyline, there's a very good chance that they're going to have an adverse drug event, and the FDA is keenly aware of this and has put a warning on the drug suggesting genetic testing for this drug. It's not required, but it's highly suggested by the FDA. And then finally, our last phenotype here is ultra-rapid metabolizer, and now we see that kind of drug level in pink here has gone way down. It's much lower than expected. And these individuals have more than two functioning alleles. And what this means is that one of their parents has what we call a duplicate allele, and they make more enzyme than what we call a quote normal metabolizer. So for active drugs, there's a potential lack of efficacy where the drug level may decrease, so an increase of the dose may be appropriate. And for prodrugs, potential risk of toxicity. So interestingly, for the ultra-rapid metabolizer, when they take a prodrug, an example of that would be codeine. If they make much more of the enzyme than normal for 2D6, they would get much more of the active metabolite in the bloodstream, so an alternate drug may be appropriate. And again, in the case of codeine, the outer shell, codeine's a prodrug, the outer shell is called codeine, the inner shell of it, or the inside, is morphine. And for the individuals who are ultra-rapid metabolizers, they would be exposed to much more morphine than wanted. And for very young children, this could be potential risk of death. Like they've noted that mothers who are ultra-rapid metabolizers who are breastfeeding send much more of the active metabolite morphine through the milk to the child, and that has been the cause of death. And the FDA has acknowledged this and put black box warnings, a black box warning on codeine suggesting genetic testing for the 2D6 enzyme. And here's our ultra-rapid metabolizer. Again, this would be for an active drug. For nortriptyline, they would be exposed to much less of the drug. So for a 2D6 ultra-rapid metabolizer taking an active drug like nortriptyline, they would get very little medication efficacy. The drug would basically be out of their body after not much more than 24 hours. So this patient would go back to the doctor and say, hey, this antidepressant you're giving me, it's not working for me. And that would be what we would expect to hear from a 2D6 ultra-rapid metabolizer taking an active drug. And here's where it gets tricky. A single person can have different combinations of these results. So for one individual, metoprolol could be potentially a big problem for them. The drug could accumulate a great deal and potentially be toxic to them. But they could take Prilosec and have a lack of efficacy at standard dose. They could take Motrin and it would probably give them good pain relief and they'd have no problem with it. And then Advair would potentially increase or warrant monitoring for adverse effects. So depending on the individual and how they're functioning for each of these different enzymes, they can have, one drug that, makes them feel terrible and another drug that doesn't seem to work for them at all. So you keep in mind, for each of these enzymes in the body, for those that have genetic variability, an individual can have very different responses. So it can be, drugs can be a little bit of a roller coaster ride for them where they can get all sorts of different response depending on the enzyme and in their genetic capacity to make a different level of that enzyme. So when approaching pharmacogenetic test results, a general rule is identify, there's free online drug gene tables. So you don't have to pay for this. You can just kind of do the math yourself and you figure out, okay, if I don't make this enzyme, then there's certain drugs, and I'll show you where to find these gene drug tables that are free online, you'll be able to figure out basically which drugs to avoid. And the reason I'm discussing this is because, and we'll get into this later, that physicians have, for the most part, a pretty limited understanding of pharmacogenetics, and it's really important that you start to understand it yourself, because you're going to, much like having Ehlers-Danlos syndrome or mast cell, you might be the one playing a role a little bit in the education of your physician on pharmacogenetics in relation to your body, and that if you get tested, that this is something you'd want to do. So you can use these online gene drug tables, which are free, and identify the enzymes you have functioning differently than normal, and then basically avoid the ones that use that enzyme. And the good news is there's almost always an alternative drug available that does something very similar or the same thing, nearly, that uses another enzyme that you will have functioning as normal. So it's basically just a game. In a way, you're playing, you're working with the enzymes functioning differently than normal and working around the ones that aren't. And so here are some of those locations where you can find those gene drug tables. The FDA has one, Mayo Clinic has one, Indiana University has one. Theirs is excellent. I'll say ours is excellent on our website too. Obviously, come back to these in the recorded version that EDS Awareness will put up, because I know it's hard to copy and paste these now, but these will be available on the online video. And these are excellent free resources from each of these websites. And it gives you an idea. We have very significant medical community that are very aware of how significant pharmacogenetics play a role in drug metabolization, from the FDA to Mayo to universities to a lab called Genelex. And then beyond that, that our lab makes available is the UScript Clinical Decision Support Tool. And I'm going to show this towards the very end, but this will just give you a quick overview of what that is. So it allows you to use basically our website, a software program on our website, to look at gene-drug interactions as well as drug-drug interactions. This is what your pharmacist does ideally when you're prescribed a new drug. Hopefully they check it in a gene or a drug-drug interaction software to make sure it's safe against your other drugs prior to handing you that pill bottle. And then the software also looks at drug-drug gene interactions, and that's where there's two drugs interacting, but also a genetic component. It also looks at cumulative interactions, and that's very important for polypharmacy patients. The average Ehlers-Danlos patient is taking, that's been tested with our lab, takes 10 medications, and that means there's a lot of opportunity for cumulative interactions where there's multiple drugs kind of stacking up on each other on these enzymes, which can create adverse drug events. And the software will identify that, and that's what makes it particularly special. So it's looking at the gene component, the drug component, as well as the cumulative component of all these drugs and genes together. There's a fee for it. It's basically $24 a month for a single patient. So about $2. I goofed on that. It's $24 a year. So it's about $2 a month. You just pay the $23.95 per year. I goofed on that. So $24 a year. And again, most importantly, if you're taking multiple meds, it allows you to become your own kind of hub for checking these interactions. It's particularly important for patients who are polypharmacy, taking multiple meds, who have multiple prescribing doctors, especially when you don't have one doctor who's really the hub for everything. If you don't have that one doctor who's asking you what your other doctors are prescribing you and checking for those interactions, then the software can really be of help to you. And you don't have to have a genetic test to use the software. You can use this now with our lab just for your drug-drug interactions, but it's going to be particularly useful if you've been tested as well, and you can enter your phenotype test results and look at gene-drug and drug-drug interactions. So as daunting as this may seem up front, pharmacogenetics is definitely very pragmatic. I like to think of it as a challenge. The test is not going to fix your enzymes. It simply identifies your genetic baseline level for creating these enzymes, and then the results of this are deemed your phenotypes. So your test results we call your phenotype. And then you and your physician work in accordance with and or around the level of enzymes your body makes or your phenotypes that are functioning differently than normal. So in a way, it's a lot like checkers or chess in that you now have the data in front of you, so you can figure out, okay, here are the drugs I need to avoid, and here are drugs that are probably going to be okay for me as far as my genetics are concerned. But then we also need to consider inducers and inhibitors for potential drug-drug interactions, which we'll get into soon here. So again, pharmacogenetics is a piece of the puzzle. Sometimes it's the answer, sometimes it's not. There are a lot of different things that can play a role in how you respond to meds. Again, like if you have mast cell, you can have allergic reactions, but it could be fatigue, pain, medication side effects, other medical conditions, diet and exercise that play a role in how you respond to meds. So pharmacogenetics is certainly a piece of this, but it's not an absolute answer to how you're responding to drugs. For some people, it's going to be extremely helpful. For others, your results may come back as normal and just be a stepping stone to what else is maybe going on. So that was our kind of 15-minute crash course, and I think I went over 15 minutes, so thanks for bearing with me. Now we'll get into the kind of nitty-gritty details about pharmacogenetics and drug-drug interactions, how to approach your doctor about pharmacogenetics, things like that. So next is what are these enzymes and how did pharmacogenetics come about? So these enzymes are really incredibly interesting. They are present in nearly all living things, so animals, plants, fungi, protists, bacteria, and viruses. So pretty much almost every living thing has these enzymes. This is a very common thing found in almost all life. These enzymes allow for detoxification of xenobiotics. But xenobiotics, that's a very fancy term for something that the body is not familiar with. For the most part, that's what that means. So when you take a drug, you're putting something in the body that the body's not going to be familiar with, especially the first time you take a drug. It's not something made by the body. It's something that comes from the outside. Like, frankly, even a food could technically be called a xenobiotic because that's not something that's made by the body. It's something new that's coming into the body. And drugs are certainly that way. And these enzymes metabolize drugs and environmental toxins as well. So something you can breathe in the air could potentially be a toxin, is then metabolized and able to leave the body as well. So, which I didn't even know about till fairly recently, and it was actually a patient who initially told me about that, and then I found out more about it in studies. So it's pretty amazing what our bodies, how much they can help, and really helping us get rid of garbage that we breathe in and that we sometimes put inside our body too, for better or worse. Interestingly, this goes way back into evolution. These enzymes are in plant-animal warfare, and there's actually a study online about that. So an example of this is gardeners and farmers will put narcissus, which are dandelions. You'll see them come up in springtime. They'll put them around certain plants where they don't want gophers, rabbits, or deer to get into it, because the bulbs are in narcissus or dandelions are toxic to animals, and those animals can't metabolize them, so they will avoid them. So it's really kind of a neat thing that you actually have farmers through thousands of years of evolution, using these plants to their advantage because they know that they're going to be toxic to certain animals, and that will keep those animals out of other places they don't want them to go. And so here's the development of pharmacogenetics. It really kind of started in 1931 when I don't have the first names here, but Girard anticipated the existence of pharmacogenetics in a book called Inborn Factors in Diseases. He basically suggested enzymatic defects leading to inborn errors in metabolism in regards to food, toxins, and drugs. And he was right. This is before genetic testing was even feasible. So he was the first to publish something that anticipated pharmacogenetics. And I'm just going to kind of jump forward here, though. Interestingly, the one I found the most interesting was in the 1960s. Vessel showed remarkable similarity of disposal, right, or the ability to, frankly, urinate out drugs for several drugs in identical twins who share 100% of their genes. So, in other words, identical twins, their genetics are 100% the same. So, if you have identical twins and they're poor metabolizers for 2D6, they are both going to be poor metabolizers for 2D6. Whereas you could have fraternal twins who you could have one who's a poor metabolizer and the other who's intermediate, and see a different response in fraternal twins taking the same drug. And so that one's really interesting to me because that allows them to really start proving what Girard was talking about way back when. And then most importantly here in the 1970s, they discovered the 2D6 variation. So now we're able to start doing genetic testing. And then in 1990, we had the Human Genome Project came online, and that's when this just took off. Genetics have just gone from 0 to 60 amazingly. It's really been 25 years since that started. And then in 2000, just 10 years after the Human Genome Project went online, the first pharmacogenetic testing labs began providing testing to physicians and direct to public. So it's pretty amazing from the discovery of this enzyme, basically 30 years, all of a sudden you can go online and buy a genetic test. And that's pretty wild, that just how quickly this happened. And again, pharmacogenetics has been with us since the beginning of human beings. This is in plants, this is in animals, it's in bacteria. And really, in the last 80 years here, did we even begin to recognize that there was a potential for something called pharmacogenetics, that the genetic variations could play a role here. And this is in regards to millions of years of evolution. Only in the last 80 years have we gotten to this point. So it's fantastic that we're able to take something that human beings have been dealing with for millions of years, and now here we are, and we can figure it out ourselves with an online test. or I should say a test we can purchase online, not do online. So now we'll jump into alleles. Alleles are found through genetic testing. They determine your phenotype, again, poor, intermediate, normal, ultra-rapid metabolizers like we discussed. The alleles are passed down from parents to children at birth. They determine your enzymatic capacity to break down drugs. So if you're familiar with what a family tree looks like, the male would be a square and the female would be a circle. And if we had both parents if they were intermediate metabolizers, then their children can have a combination. If they have 3 kids, feasibly they could have all normal metabolizers or one intermediate or a poor metabolizer or any combination of that amongst their kids. So in other words, it's genetic, but it's also random how it's passed down. If both parents were normal metabolizers, then all the kids are going to be normal. If both parents are poor metabolizers, all the kids are going to be poor. If both parents are intermediate, then you're going to get kind of any combination of result. If both parents are ultra-rapid, all the kids will be ultra-rapid. So again, it's genetic, but also can be random at the same time depending on how the alleles pass down. And again, don't worry too much about this. This is getting into the technical part of it. For those interested, that's what's happening here. And this is what it looks like on a result. The alleles are typically reported in a format of star number / star number. An allele is received from your mother and one from your father. Together, the 2 alleles help determine your phenotype. Some individuals receive duplicate alleles from a parent, which can affect the outcome of their phenotype. Generally, as a patient, you really don't have to worry too much about the alleles or the genotype. It'll be most helpful for you to discuss the phenotype with your doctor. Your doctor really is not going to know the alleles by far and wide. It would be rare for a doctor to understand what the alleles stand for. What they're going to understand better is the phenotype, one of those 4 test results. This is a very busy slide, but just to give you an idea in the population, and I'll just focus on 2D6 here. For 2D6, about 5% of the population are poor metabolizers. So these are the individuals, again, we worry the most about. These are the folks where if they take an active drug, there's a very good chance for drug accumulation, and depending on the drug and how dependent the drug is on the enzyme, potential for toxicity. Interestingly, about 36% of the population, right, that's a huge amount of the population, are intermediate metabolizers. That means they make about half the normal amount of the enzyme. And then about 3 to 4% are ultra-rapid metabolizers. So when you add that up, for 2D6 alone, it means about half the population, 50% of the population, is not making a normal amount of that enzyme, and the other 50% is. So that means for 25% of all drugs on the market, half the population is going to respond to that drug differently than anticipated. And for the intermediate metabolizers, it may not be so much that it's going to be a major problem. They might not even notice. But if you're poor metabolizers or ultra-rapid metabolizers, depending on whether the drug is an active drug or prodrug, that drug could potentially send them to the hospital or just have a complete lack of efficacy or a lack of efficacy for the most part. So that's really significant. The 2D6 enzyme can affect how a great percentage of the population responds to medications. So next we'll jump into the body as a constant cycle of experiment. This is not going to be super technical as far as this overview. What I want to just review is just to give you an idea, if so far this has been a little bit technical for you, I want to talk about this in another way of how we look at our bodies. And one is how you would feel when you eat certain foods or drink certain drinks. And depending on, frankly, on just, the family you grew up in and the foods you ate in your family, I know for us, we always had soda in the fridge, and I was always drinking caffeine-free soda with tons of sugar in it, and I had really no realization on how much that sugar was affecting me as a kiddo because it was what I was used to drinking in the family. So if that's what you know, you don't know any better. And it's important for us to consider literally, Buckminster Fuller would talk about you are what you eat. And we need to understand when we eat food, that food, after X amount of days, it literally becomes our skin. Our body's constantly regenerating. So the foods we eat have an incredible impact on our own health. And so, if we're eating what's pictured here, we're kind of eating junk food, sugar, processed foods that mostly come in a box or a bag. Our body will be responding to that, and it's not going to be, the most ideal circumstance. And so, if you're eating, fruits, vegetables, barring any allergies, forgive me myself, folks, because I know there's certain foods that you definitely need to avoid for your health. But barring allergies, you're going to feel much, much better eating fruit and vegetables than you would be, getting something in a box or in a bag. And I just know this personally because that's been my own life experience too. And different bodies work differently. And some people, it makes more sense for you to eat meat or things like that. But for one thing we know is avoiding what we kind of deem junk food is a really good move. And especially if you have a chronic condition, getting your diet right is the number one most important thing you can do as far as I'm concerned. And then working with your doctor for the rest. So, keep that in mind. What you put in your body absolutely has an impact on and how you're doing in general and how good you feel. And again, another example, how do you feel when you drink water versus wine or versus soda? Barring that you live in Flint, and there's problems with the water, which we're learning about, in various places, so we can't even really say the water is always the best thing, but, it's definitely going to be a better alternative to soda or alcohol, and just something to keep in mind if you're kind of a regular soda drinker. It's going to be, water is going to be a much, much better alternative to a sugar drink for the body for the most part. And then finally, to keep in mind that, drugs, every time you take a new medication that you've never taken before, you are running an experiment. You don't know how you're going to respond to that drug. You could have an allergic reaction. You could have no reaction, and the drug works perfectly for you. You could have an adverse drug event because of genetics, or the drug could interact with something else. And so it's something to keep in mind, and we certainly don't want to be afraid of drugs because drugs are incredibly important to sustaining human life. There's no question that, I can speak for my own father. He may not be here today if it wasn't for the drugs he's taking right now. And it's important for us to keep in mind that there's a quality of life where it really is important for us to take medications. And I want to be very clear that at no point do I want to not advocate for taking drugs when it's appropriate to take drugs, because it certainly is important to do so when the timing is right and when the condition's right. I just wanted to show a slide. It's actually from an Ehlers-Danlos Awareness Center EDS awareness slide that Dr. Pasinki gave, and I think that's kind of ties it all together. Dr. Pasinki is really good about focusing on the cycle where if you're in pain, your sleep is affected, and then if your sleep is affected, if you're not getting enough sleep, that can lead to fatigue. The fatigue can lead to depression, and then you end up in this cycle. And that's why I want to stress that there are times in our lives where taking a pain drug is definitely the right choice because it's going to affect our quality of life, and it's going to keep us from having a lack of sleep, which could lead to, again, to fatigue, depression, things like that. So getting a good night's sleep, if taking pain medication allows for that to happen, it's really going to be, probably the right choice for the individual. So keep that in mind as I go along here. I don't want you to be afraid of drugs as we talk about adverse drug events. I know it can sound scary, but there are absolutely times where taking medication is definitely the best choice, no question whatsoever. So I don't want you to think otherwise as we go on here. So next, polypharmacy and pharmacogenetics in the Ehlers-Danlos community. So these are typical medications prescribed that we've seen in the Chiari hypermobile EDS population that are dependent on these enzymes we're discussing. And you'll see a lot of things you've probably taken, right? Oxycodone, tramadol, bupropion, which is Wellbutrin. These are the generic names: amitriptyline, hydrocodone. So really, these are commonly prescribed drugs that are prescribed to people. Ibuprofen, NSAIDs. These are prescribed every day to hundreds of thousands of people around the world. And all of these drugs are dependent on these enzymes to one degree or another for their metabolization. So specifically within the Ehlers-Danlos community, of the hundreds of patients tested who have Ehlers-Danlos, who tested with our lab, we see on average that they take 10 medications. That's from our own internal population data from 2014. And then from an Ehlers-Danlos awareness published study just in May of this year, they found that 87% of patients with Ehlers-Danlos who responded to the study have 3 or more healthcare providers. So that leads to a question of polypharmacy. If you have 3 healthcare providers and they're all prescribing meds, there's a potential that those doctors aren't talking to each other, and that there's potential for drug-drug interactions when that happens. And again, that's a large percentage of the community, 87%. And then the most common reasons for ineffective pain management is attributed to difficulty finding medication that works. So 61% of survey respondents said that. And then having reactions to medications they try, which is 52%. And again, this is from the EDS awareness study published in May. And so certainly pharmacogenetics and polypharmacy are going to play a role in your own survey results. And so this is, to give you an idea, just of polypharmacy, for the moment, we're going to take the genetics out of this. And this is a slide that shows the number of potential interactions. I'm going to put my cursor on here. So here's the potential of number of interactions from 0 to 200, right, going up, depending on the number of meds you're taking. And that going this way is 0 to 20 meds. So in other words, if you're taking 20 meds, there's a potential for almost 200 just drug-drug interactions, not even considering the potential for genetic interactions. And so for Ehlers-Danlos patients taking on average 10 meds, we see that there's going to be a potential just for drug-drug interactions for 40 of them. Now, it depends on the medications being taken. And again, this is taking it to the extreme. This would be, odds are we're not going to have a patient taking 10 meds having 40 drug-drug interactions. It's an extreme example. But I also want to give you an idea, if you're not already familiar with polypharmacy and the potential for drug-drug interactions, that there is a reality where there's a potential for drug-drug interactions that could be very significant. And it's not something to worry about, but it is something to be aware of, just that it's in your mind. And each time you take a new medication, that you really want to check with the pharmacist and make sure that that new drug is not interacting with other drugs you're taking. And I want to stress, in a busy pharmacy where it's a chain that is particularly busy in a busy city, where the pharmacist, you hand them the prescription, they turn around and hand it back to you real quick. Make sure they stop and take time. And I would ask them every single time, point blank, I don't care how busy they are, say, hey, I'm taking a lot of meds. Can you check to make sure that the new medication you're handing me today is not interacting with every single one of my meds? And make sure they have a list of your supplements and any herbals you're taking too, because they'll have software that can check for supplements and herbals as well. And we're going to talk about supplements and herbals soon, and you'd be surprised that some very commonly taken popular supplements and herbals that popular people, medical professionals on TV talk about can really throw a wrench in and interfere with some other meds. So it's really important to have the conversation with your pharmacist and really kind of hopefully start that conversation with them if you already haven't, and try to create a relationship with them so that they stop and take that time with you each time, no matter how busy they are. That is their job. I don't care if there's 10 people behind you. It's their job to make sure you walk out with drugs that are not going to interact in a negative way together. So now we'll talk about inhibitors and inducers. And again, this is just drug-drug interactions. We're not talking about genetics here yet. And I love this slide because it just is a perfect example of, these drugs that look so innocent, yet here they are. potentially one of them being a culprit. And actually, these pictures here were actually suggested by a patient at the Mast Cell conference a couple years ago. Hillary, if you're out there somewhere, thank you. And she suggested using kayaks as an example. And so I'll jump into that. But think of a kayak as the drug in this case, and then the water as metabolism. And moving forward with the photos, I'll show you all even examples of that. So here's what we want. We want the kayak on beautiful, calm water. Kayak is the drug, the water's metabolism, right? This is what we want. That's what we're shooting for. But sometimes we're given an inhibitor. And this is probably a pretty good example of what that might kind of look like in the body. The water is, you could consider it as something that's slowing down the drug or the kayak, right? Potentially even stopping it, where the kayak can't even move forward because it's in this icy water that has obstacles. So inhibitors can be in the form of drugs. So typically it's going to be in the form of a drug. In other words, you're taking a drug, and it just so happens that drug interferes with enzymes in the body. So it can be a drug, it can be an herbal, it can even be a food, and it can even be a breathed-in chemical. If you're working in a factory with a lot of chemicals, it's feasible that something you're breathing in can be an inhibitor and interfere with your meds. Inhibitors compete with other drugs for a particular enzyme, thus affecting the optimum level of metabolization of the substrate drug, which in many cases affect the individual's response to that particular medication, making it ineffective. And I'm going to show you what some of commonly prescribed inhibitors are moving forward. But just kind of keep in mind, the inhibitors basically are keeping your body from making the specific enzyme they inhibit, and thus can affect other drugs you're taking that are dependent on that same enzyme, and that thus can lead to drug accumulation or toxicity. And again, it's going to be very similar to what we would call a poor metabolizer, right? Where there's no inhibition, the individual, takes a drug, they metabolize the drug is urinated from the body, it leaves the bloodstream. But for someone taking an inhibitor, an enzyme inhibited by another medication or substance, it leads to accumulation in the body of the active drug, which is dependent on the now inhibited enzyme. So in other words, you could be what we call, quote, a normal metabolizer genetically for, let's say, the 2D6 enzyme. But then you could take a drug that inhibits the 2D6 enzyme, and that basically renders you, or we call it phenoconverts. It changes you during that time to a poor metabolizer. It changes you to someone who doesn't make that enzyme while taking the drug that's inhibiting that enzyme. And thus, if you take any other drugs dependent on that enzyme, that drug's not going to be able to be urinated from the body. It's going to stay in the body, and it's just going to kind of loiter in the body. And if you continue taking that drug, it's going to continue to accumulate and potentially become toxic in the body and lead to an adverse drug event, or depending on the drug, hospitalization even, the worst case scenario. And inhibitors. And that link I gave for Indiana University has a list of inhibitors. So the cool thing is right after this, you can jump onto their website for free and go look up any drugs and see if you're taking any drugs listed as inhibitors. And I just circled these because here are some commonly prescribed inhibitors. Bupropion, again, that's Wellbutrin. Fluoxetine, that's Prozac. All of these are inhibitors of these different enzymes. But the cool thing is all this is online. You can actually just look it up. You don't need to pay anybody to look at this list of drugs. And the ones I want to discuss today, because these are the ones we see that are the biggest problems. So powerful 2D6 inhibitors we see on medication lists coming into the lab from patients who are tested with our lab are listed below. These medications and herbals inhibit to varying degrees about 25% of meds on the market, so a quarter of meds on the market. So in general, I would say would be great to memorize these if possible, or keep these in your back pocket, but keep your eyes peeled for these when taking multiple meds. These are responsible for hundreds, if not thousands, of adverse drug events every single day in the United States and around the world, and continue to be prescribed by physicians and naturopaths who are unaware of the powerful enzyme-inhibiting effects of these commonly used meds and herbals. And look at this, Prozac, right? The Sopranos probably made Prozac very famous, right? The antidepressant that Mr. Soprano was taking, right? It's probably the world's most famous antidepressant. And a reason, in part, that Prozac, or fluoxetine, the generic name, gets such a bad rap is because and a lot of people just don't know this. Part of the reason is because it was a 2D6 inhibitor, and it was interfering with 25% of meds on the market. So if you were taking another drug, that was dependent on the 2D6 enzyme, Prozac would make that drug level, depending on how dependent it was on the enzyme, accumulate. And I guarantee you, there's been hundreds of thousands of people who've ended up in the ER because of Prozac for that reason, because it was interacting with another drug. And to be very frank, a lot of physicians are not familiar with this. They don't know about this. So we as patients, especially if we're taking multiple meds, it's really important that we start to pay attention to this on our own so we can be our own advocates and just be aware of this happening. So Prozac, Wellbutrin, Cymbalta, Paxil, all four of these are very strong, powerful 2D6 inhibitors. Another one is rhodiola. That's an herbal supplement. We see a fair amount of people taking that. You can get it, I think Dr. Oz is one of the folks, or one of the kind of major medical folks on TV that talks about herbals a lot, will advocate for rhodiola. And by itself, no problem, right? Genetically, it can't interact with something else if you're only taking rhodiola by itself. But if you're taking another 2D6 medication, it could potentially make that other 2D6 medication increase in the bloodstream and create a problem. And we've seen this on the list of Ehlers-Danlos patients. I'd say if you have EDS in your polypharmacy, you're taking multiple meds, you really want to double check whether you want to continue taking that and speak to your doctor or naturalist or about it before changing it, because it may interact with your other meds. They may already be aware of that and dosing things accordingly. But, it's something you don't have to get with a prescription, so that's why I really want to point that one out. And then another one that doesn't affect 2D6, but we see it a lot, is turmeric. Inhibits 2C19, 2C9, and 3A4 moderately, and we've seen that could really, when I say throw a wrench in, right, it's interfering with a lot of other medications potentially on the market. So those are the ones we see a lot, and I just want you to be aware of, because odds are, a quarter of the people on this call, or probably half the people on this call, have taken one of these things before, and potentially even experienced adverse drug reactions because of these without knowing that these were the culprits. Again, what we want, the kayak is the drug, the water is the metabolism, and now we're going to talk about inducers, and this is another type of thing that we can get in a drug or other thing. So in this case, we see the kayak is going through just a massive amount of water. And again, this is like a drug going through someone who makes tons of the enzyme, and the drug just goes right through the body. They really don't hold the drug in their bloodstream. The drug is leaves the body very quickly through urination because they make so much of the enzyme that the drug just sails right on through. So an enzyme inducer is a type of drug, herbal, food, or chemical that increases the metabolic activity of an enzyme either by binding to the enzyme and activating it, or by increasing expression of the gene coding for the enzyme. And then, again, this is a free list on Indiana University's website. I gave or actually it's right down here to the link, and you can go on here and look at inducers. And one of them that I've circled here that I want to briefly discuss, because we see it commonly here, is St. John's wort. So you can buy St. John's wort without a prescription. You can walk into any grocery store or pharmacy and get it. And St. John's wort is a 3A4 inducer. And what that means is it makes your body make more of the 3A4 enzyme. And the reason I bring that up is because there have been studies that have shown that, so I'll give you an example. Let's say you have a young woman who doesn't have insurance, and she is depressed, and her friend says, I've heard that St. John's wort is kind of a good natural antidepressant. You don't have to, pay someone to prescribe it to you. You don't have to see a psychiatrist or go to your doctor. You can just buy it and take it. And if she's taking estradiol, which is commonly found in birth control, the St. John's wort, and it's dependent on the estradiol is dependent on the 3A4 enzyme to be metabolized by the body. If she was taking St. John's wort at the same time, it would make the estradiol have less efficacy. In other words, she would be more, the birth control pill wouldn't work as well in the body, and because of that, she would be at risk of potentially having an unplanned pregnancy. And there are studies that show this. So this is a commonly known interaction. And again, this is something you can just buy in the store, and we know that this is happening, to God knows how many people. This combination that they're just unaware of. So we really want to be conscious of, again, when we put something in our body, what is it potentially going to do? What are the possibilities that will happen here before I take this drug or this food or what have you? Again, mainly we want to focus on drugs, but it's really to keep in mind everything we put in our body is potentially going to make things better or worse, and we want to be conscious of that before doing so. So pharmacogenetics in the Ehlers-Danlos community. So now we're going to look at genetics in the Ehlers-Danlos community. So we looked at 203 patients who had Chiari, hypermobility, and or Ehlers-Danlos syndrome, tested in relation to 21,000 general patient population tested with Genelex. So that 21,000 folks, those are all people who, for one reason or another, said, I think I need a genetic test because I've had a poor response to drugs in the past. And so we don't want to say the general world population or U.S. population, but we do think that that number probably actually correlates very well with the general population anyways. So this study, it's not I should say it's not even a proper study at all. It's just correlation of data. So treat this data as informal. Frankly, you can take it with a grain of salt. But because we had it available, I wanted to show you to give you an idea of what we believe to be the case, just looking at correlation data. So this is from Dr. Fraser Henderson, who was very kind to allow us to use this data from his Metropolitan Neurosurgery group. We looked at about 100 of his patients, and the patients in orange are going to be our, Chiari hypermobility group. Most of them probably have Ehlers-Danlos syndrome. And then the blue are the U-script patients. And basically what we did is we just said, okay, how many of these patients are normal across the board? And interestingly, 7%. And that number correlates, perfectly with what we see in the general population, that 7% of the population had all 5 of the enzymes functioning as normal, and that went for the 21,000 who have tested with our lab, as well as the 203 patients who are in the EDS-related community. And we see all the other data very similar. It's off by just, no more than like 2%, or maybe, about 2%. So when we correlate that data, just with his patient group, we can say, we don't see that the Ehlers-Danlos patients have more of these enzymes functioning differently than normal than other patients who've been tested with our lab. And so then our next question is, okay, well, that was one office. What if we look at a second office as well? And so we looked at Dr. Posinki's patients at the time with George Washington University Hospital. And again, he was very kind to allow us to correlate his patient data with folks who've been tested with our lab. And we see it differ a little bit, but again, it's pretty darn similar. We see maybe a 6% difference, but it's not too far off from what we saw before. And that's something to keep in mind that in both offices, when we looked at 203 patients, it was very similar to the general population who had tested with our lab. So I feel personally pretty comfortable saying, based on the correlation data we have at the moment, it's fair to say the patients with Chiari, hypermobility, or EDS are not more likely to have these enzymes, the cytochrome P450 polymorphisms, for these 5 enzymes we looked at. We just looked at these 5 than any other patients tested with Genelex. Now, very importantly, that said, there may be other enzymes and are likely other factors that have already or have yet to be identified that account for increased amounts of adverse drug reactions or lack of efficacy in patients with Ehlers-Danlos syndrome. So basically what I'm getting at there is when I talk to physicians and what I've heard from physicians is very often they say, they see in the EDS community that EDS patients are responding very differently to drugs than the general population. And as far as I believe, just with correlation data, I don't believe that has to do with the EDS community being any different as far as these cytochrome P450 enzymes. I think these enzymes are probably going to, barring ethnicity, which plays a role, but kind of across the board in general, I think these enzymes are not going to be any different for EDS patients compared to the general population. And my guess, and again, I'm not a doctor, I'm not a pharmacist, I really have no business saying it, but my guess, and I think a lot of folks who are in the Ehlers-Danlos community, both patients and medical professionals, is that there's something else related to EDS or one of the related conditions that is also playing a role with the potential for having different response to meds. And we see that with like lidocaine and novocaine. We know that seems to be kind of specific to hypermobility or ADS. So we kind of already know that, but I don't want to be controversial about it at the same time. But the main thing I want to point out is, as an Ehlers-Danlos patient, I wouldn't say run out and get a pharmacogenetic test. If you've never had an adverse drug reaction, you probably don't need a pharmacogenetic test, assuming you've taken multiple meds. If you've never taken a med, I would say you don't need to, once you take a med, and this sounds bad, once you take a med and you have a bad response, they say, yes, it makes sense to pay money to get a test done to figure out what's going on. And if you're rich, wonderful, get the test done prior to, having an adverse drug reaction to allow yourself to see that potential. But I'd say, if you're not having any problems with drugs, don't go take a test for it. And I don't see a reason for Ehlers-Danlos patients to run out and get this test done any more than anybody else in the general public, because I don't see that it's affecting other individuals anymore. Again, just as far as these cytochrome P450 enzymes are concerned. So next, pharmacogenetic landscape, approaching and ordering testing. So an anonymous cross-sectional national pharmacogenetic survey of over 10,000 physicians was conducted in 2012. Here's the good news. 97% of the responding physicians agreed that genetic variations may influence drug response. And that's great news. That means, you walk into a doctor's office and 97, it's almost correlated to belief in global warming, right? 97% on the right side of font science here, and they say, yep, I understand that genetic variations potentially play a role in drug response. And that is the right answer. The bad news, only about 10% felt adequately informed about pharmacogenetic testing. And so this is what we're going to talk about in this next section here, the pharmacogenetic landscape, and really, where doctors lie in this, because this is a question almost every single patient asks. They say, my doctor doesn't understand this. I don't know what to do. How do I talk to my doctor? This is the way I like to think about it as an analogy. Imagine that your doctor is a landscaper. So you hire a landscaper to, come and trim your trees and your bushes and your lawn. In landscaping, you typically are not going to have a landscaper who specializes in bonsai, right? That is landscaping, but most of your landscapers are not going to know bonsai. Maybe, I don't know, 3%. I have no idea what the number is, but a small percentage are going to be experts in bonsai. And so when you go to your doctor and you talk about pharmacogenetic testing, it's kind of like going to your landscaper and saying, hey, I want you to do bonsai. And they would say, well, bonsai is a really specific study, and I haven't done that, and, I would need training on that before I'd feel comfortable going into your yard and cutting your trees in the bonsai fashion. And I think that's a weird but decent analogy for bringing pharmacogenetics to a doctor, because I think what's happening is they're saying, that's not what I do. I was trained to diagnose patients with a condition, and what you're talking about is something that is, not a disease condition. It's something that is more a branch of, it's pharmacology. It has more to do with what a pharmacist might do. The problem is pharmacists cannot prescribe a genetic test in the United States. They can, in other words, someone who can prescribe a genetic test is someone who has to be able to prescribe a medication. So we can't go to a pharmacist and say, I want a pharmacogenetic test. Can you sign this and send it to the lab for me? A pharmacist cannot do that. They can help you with the process, but they can't actually order the test. It has to be a doctor. And so the pharmacogenetic community has created this conundrum in the industry for doctors, because now we have patients, our lab says, hey, order a test kit from us, take it to your pharmacist, or your doctor rather, your landscaper, and ask your landscaper to do bonsai. and that's what we have to understand, is there's this weird dynamic, and the labs that do pharmacogenetic testing are really throwing that in the lap of the doctors, and we cannot blame doctors for that. They have not had the opportunity to learn about pharmacogenetics. They haven't learned, it's like, again, a landscaper who does bonsai, and we suddenly say, hey, I know you cut my yard this way for years, but now I want you to do it a completely different way in a very specific Japanese style that is, odds are you've had no education about whatsoever. So moving forward, when we think about this, we really have to advocate for the doctors in knowing that we are throwing a curveball at them, and that that's their reality, and that we need to empathize with that fact. And I was so happy to read about that in Dr. Lawrence Safran's book, Never Bet Against Occam. In the back of the book, he has, I think, 2 pages on pharmacogenetics. And he basically, I just want to give him credit, because he basically said the thing that I have not heard from a doctor ever. And I really appreciate the fact that he said it in his book. And I'm just going to read, this is a page directly from him, so Dr. Afrin, forgive me for plagiarizing here. So he says, although many medications interfere with the cytochrome P450 isoenzyme functioning, and although these enzymes, these polymorphisms, are far more prevalent in the human population than most doctors are aware, it is difficult to diagnose cytochrome P450-related problems, because it's difficult to suspect cytochrome P450-related problems. So he says, why? Principally because the number of medication and cytochrome P450 and drug interactions is vast and can't possibly all be remembered. Remember those, I'll cut in here, those drug lists I showed you before. We're talking about hundreds of drugs. There's no way anybody's going to memorize that. Back to his quote here. So principally, the reason for this is because the number of medication and cytochrome P450 interactions is vast and can't possibly all be remembered, because medical training about CYP450-related problems consists of about a minute's worth of in-class lecturing relatively early in the decade or so of training most doctors get, and with no follow-up clinical training. So in other words, this is like a speck on the radar of physicians. And now these days, okay, maybe medical school you're going to learn more about it because remember, in the 1970s they discovered 2D6, the 2D6 enzyme, and it wasn't until the 1990s that we were doing the genome project. So let's face it, a doctor in his 50s or 60s, this was not taught in school for them because it wasn't even it wasn't even really, discovered and made published in a way that they could use it in a clinical fashion. So we have to understand that the doctors cannot be blamed for not having an understanding of this, but we can help them with that. So, and then finally, he said, little wonder then that the vast majority of people with these related problems never get diagnosed as such. And I'm going to speed up. John gave me a reminder here. I'm going to speed up a little bit so we have about 25 minutes to go and still some questions. So again, genetic metabolic differences in individuals may raise uncomfortable questions for the pharmaceutical industry, for medical education facilities, physicians, pharmacists, and patients. And it's again because we've really just learned about genetics fairly recently. There hasn't been a lot of education about it. And there's even articles. This one just came out 2 weeks ago. Precision medicine may mean more uncertainty needs to be communicated to patients. And that's true. We're starting to talk to patients about, because of the internet, frankly, in ways that we never have before, and that can bring up some weird situations for medical professionals. So assessing patients for pharmacogenetic testing. So adverse responses to medications can take many forms. If a patient experiences symptoms immediately, within a few weeks, or even within months of starting a new medication, it may be an indication of an adverse drug event. These can come in many shapes and sizes depending on the drug. Brain fog, anxiety, headache, GI pain, seizure, fainting, fall, lack of medication efficacy. By the way, these are all things that are in studies that have come out showing that these are responses to an adverse drug event where the patient didn't make the enzyme or made much more of the enzyme than normal. And the problems with these symptoms, if you're looking at this and you're an Ehlers-Danlos patient, is these can all be symptoms of other medication or medical complications such as Ehlers-Danlos syndrome, mast cell activation disorder, Chiari, gluten intolerance, or other things. So in other words, it could be very complex for even the best medical professionals in their field to pinpoint an adverse drug event. And we really need to understand that as patients, that especially as complex patients who might have multiple medical conditions taking multiple drugs, there are all sorts of potential reasons why you might be having certain reactions. And it may have to do with genetics or nothing whatsoever due to genetics. And we have to understand that human beings are very complex, and we have to give our doctors some space on that, I think. And I've learned that over the years, that especially the really good ones deserve recognition and can't be expected to understand everything that's wrong with us. And this is a picture of Lee Hood, and he's here in the Washington area, and he coined a term called P4 medicine, which I really appreciate. And this is where you come in. He really recognized that a medicine is, these days, is now predictive, preventive, personalized, and participatory. And he really says because of the digital revolution, which is the internet, and then new systems biology and systems medicine, like labs like Genelex, and the data that's coming out of the Genome Project and groups like CPIC, that there's now a consumer-driven healthcare and social networks, which you're very well familiar with, social networks being an EDS awareness advocate yourselves. And they, all of a sudden, you know this as an Ehlers-Danlos patient, you're playing more of a role in your own advocacy than you ever have in the history of medicine in all of the world. So it's really a special time to be alive and to be able to do this. And to keep that in mind, that you really have an opportunity to be a big part of how medicine goes for you moving forward, and understanding that you work with your physician on this and that you can be your own advocate. And I appreciate Lee's sentiment about that. So how to approach your physician. And this is probably the best quote I know. I've developed this after a decade of talking to patients and doctors. And I would quote me. So if you go to your doctor and you're worried that they may not know about pharmacogenetics and you think you want to get tested, this is what I would say. Doctor, as you know, I've had a history of poor response to medications that we've yet to find a diagnosis for. You also know that drug metabolism is affected by genetics. I have a testing kit from a lab that I trust to perform pharmacogenetic testing. Do you think it's something we should consider together? And the beauty of this is you're not coming in and saying, oh my gosh, I have this problem with drugs. I know I'm a poor metabolizer, ultra-rapid metabolizer. I haven't been tested yet, but I know this is the case, and this is going to fix me. If you walk into your doctor's office with that, they are going to roll their eyes probably in front of you. That's not – I've just found that backfires every time. What you really want to come in and say, be very pragmatic. Say, hey, drugs play a role in or genetics play a role in drug metabolism. I've done some homework on this. What do you think? And put the power back in their hands. Let them comment on it, because that's where their comfort level lies. They're much more comfortable getting to share this with you than you coming in and saying, I need you to do this test for me. That's, I think, really the best way to word it. And for patients who have done this, like 99% of the time, the doctor's in agreement, because it's very logical and it makes a lot of sense for everybody. So to order testing, you can just ask your doctor if they have a lab they're already comfortable working with. Odds are they don't, because they haven't done pharmacogenetics before, or maybe they have, but not much. If they haven't, just call a lab and order a testing kit. There's a lab called PGXL. There's a lab called AssureX, and then GentalX, my lab that I work for. These are all high-quality labs that work that have good track records. The reason I mention these, there are definitely some labs that are not so great. They don't test for as many alleles as other labs, and that means there's potential where their test results won't be as accurate. And so I don't want to tell you to do the test with our lab if you're going to consider testing. I would love you to, but but I want you to know the names of a couple labs, so call around. Frankly, if you could call the lab and talk to a human being, that's a very good sign too. Sample collection can either be a blood or painless mouth swab test collected at home, so it's really easy to do. It does need a doctor's signature though, so keep that in mind. You're gonna need to ultimately take it to a doctor to sign off on. So cost. This is the big question. Labs charge various fees. It can vary from $150 to over $2,500 depending on the test run and whether the patient is paying up front with cash or insurance is being billed. Like any consumer product, call the lab to make sure you're comfortable with them and ask what you get for the fee they charge. It can vary heavily. Some labs will focus on just a couple enzymes. Others will test for a whole bunch or both, like our lab. We can run a 5-enzyme panel or run 24 different tests for you. Private insurance coverage is very hit or miss and can also vary from lab to lab. Medicare and Medicaid only cover the testing in very specific circumstances, and generally they don't, but they have in the past. So there's a lot of confusion about that. Medicare used to cover this across the board for all labs. They no longer do, except for like Plavix, nortriptyline, and amitriptyline. This may change in the future, but for now that's the kind of how it is. I frankly would say, call a lab and just say, hey, here's my insurance company. What's my likelihood of getting covered? If it's not likely your insurance will cover, ask them the cash pay rate. Because these days the cost of the test has gone way down because the technology is getting better and better, and it's not an outrageous price anymore. And haggle with the lab. Say, hey, what can I get for my money? Now we'll look at the UScript Clinical Decision Support Tool. We have about 15 minutes to go, so I'm going to be very quick on this. There's much more information on this on our website, so I'm just going to jump through it really quick. We went through this slide already, so I'm actually going to skip it. Again, it's $24 a year, not a month. And it's a cumulative drug-drug interaction software. That's what makes it special. We talked about that a little bit already. It's going to look at, I think right now, over 2,700 different drugs and metabolites. So it's, looking at supplements, herbals, even certain foods, even recreational drugs that are dependent on these enzymes. And it identifies more drug-drug interactions than typical drug-drug interaction programs because it includes drug-drug interactions at the enzyme level and then, of course, drug-gene interactions, which is the most important component for our purposes. So here's a quick example of what that looks like. So here's amitriptyline with a 2D6 pore metabolizer, and I'm going to add my cursor here. So our software would give it, basically a stop sign and say, this is not a recommended interaction or not a recommended drug for this individual. We see that the drug level would go up, nortriptyline, like we saw in our earlier slides, the drug level would go up significantly. We estimate 76 to 200%. Almost certainly this person would have an adverse drug event, and if they continue taking the drug, end up in the ER. Using the software, we can find the best alternative drug, and that's the beauty of it. It takes, running through this on the software takes probably about 2 minutes tops to find the better alternative drug. And for this individual, a poor metabolizer, amitriptyline wouldn't be a good drug for them, but citalopram would be as far as their cytochrome P450 enzymes are concerned. So we can literally, taking about 2 minutes of your... Perfect. And we have, this is great, we have about 10 minutes and we have 10 questions, so we'll do about a minute each and we'll cover our bases here. So the first question, how effective is the testing for determining how well a person will metabolize or respond to pain medicine? This individual says, I'm going to undergo potential neck fusion and I do not seem to respond to many pain meds. I'm hoping that genetic testing would help me figure out which drugs would actually be able to help me with my surgery so my doctors are better able to treat me after surgery. And so the answer to that is, it depends. If the issue is pharmacogenetic, if you're, for example, 2D6 and 2C9 and 3A4, those three enzymes that are make up 3 of the 5 main enzymes we talked about handle different pain meds. 2D6 and 3A4 handle a lot of opiates, not all opiates, but a lot, like codeine, hydrocodone, oxycodone, tramadol, fentanyl, and then 2C9 handles most NSAIDs by far and wide. If you're, depending on the level of the enzyme you're making, yes, it could definitely play a role in your response to pain medication. But I've also had a patient who sounded like a textbook case of not making these enzymes, got tested, and your results came back as normal. And that could be the case for you too. So it's a little bit of a gamble when you run this test because we don't know how helpful it's going to be until we get your results. And for some individuals, it can be incredibly helpful. And for others, it's just a stepping stone to something else. So the answer is it could be extremely helpful or not at all. But it's still, I would say if you have had adverse drug events or lack of efficacy with meds, consider getting tested because if it is the issue, it's going to help. If it's not, at least then you've ruled it out. A couple other questions here. Is there a test for 3A4, 3A5? Yes, there is. Next question, what P450 pathway does oxycodone go through? That's 2D6. What are the signs of toxicity? It would be the same as, taking too much of oxycodone. So it would be, similar to having opiate accumulation or toxicity. Because I'm not a pharmacist, it would be best to discuss that with a pharmacist. I would ask just, frankly, you could look it up online too, but for the sake of time, but you could ask your pharmacist or look it up online. Are there any NSAIDs that don't go through 2C9 pathway? Thank you for asking. That's a beautiful question. And I learned this from Dr. Posinki because this happened with one of his patients. Naproxen, which also goes by the brand name Alleles. Alleles does use 2C9, but only as a minor pathway, and he's found for a patient who is a 2C9 poor metabolizer, ibuprofen was not good for them as an NSAID, but Alleles or naproxen was. So I feel very safe saying to a 2C9 poor metabolizer that if you're going to take an NSAID, that naproxen or Alleles will be the right NSAID for you. Next question here. I've heard that 23andMe tests for the CYP enzymes. Do you know if this is true? Yes, it is true. Yeah, and their support is not great. I'm not a big advocate of 23andMe because they give you the raw data, and then it's up to you to find a middleman company that translates that data. And we've had cases where a patient got a different phenotype from the middleman than our lab, and then kind of came to our lab and said, hey, Genelex, you gave me the wrong result, and we never like to hear that because if we've yet to find it. We are very serious in what we do. We have an incredible lab and very talented people, and we ultimately figured out, it was the middleman company that gave them the wrong phenotype information. So I'm not a big advocate for 23andMe, and that's why I would suggest one of the other labs I mentioned earlier. Next question, for poor serotonin transporter phenotype, will tramadol have a poor outcome because it raises serotonin levels? Ooh, I don't have an answer on that one. That's going to be a question for a pharmacist. I don't actually know in regards to tramadol and the 5-HTT or the serotonin transporter. Next question, if you are on one of these powerful 2D6 inhibitors that we talked about earlier and have had UScript done or the Genelex testing done in the UScript software, and your results show that you're a poor metabolizer, could your body learn to remake the enzymes needed to metabolize the medications that are metabolized by the pathway? If you were a poor metabolizer, the answer is no. Once a poor metabolizer, if that's your genetic test result, then always a poor metabolizer. My understanding is if you were to take an inducer, it wouldn't have any effect because if you weren't making any of the enzyme, it wouldn't do that. Or if you make very little of the enzyme, maybe it could do it a tad bit. But I would say just avoid, if you're a poor metabolizer for 2D6, I would say try to avoid 2D6 drugs entirely if reasonable. Again, don't make any changes without talking to your doctor first. Next question, if you're a poor metabolizer in several pathways shown by UScript or any lab, right, can you have immediate reactions to the substrates listed? Yes. Yes. If you're a poor metabolizer and you take an active drug that's dependent on that enzyme for metabolization, depending on how dependent that drug is on that enzyme for its metabolization, you could potentially have an immediate reaction, or it could build up over much time, just depending on how dependent the drug is on the enzyme. Next question. Since I picked up a packet for testing at the EDS conference, not one of my physicians felt qualified to interpret the results and therefore refused to sign off on the test. Yeah, sorry to hear that. It can happen. My PCP finally agreed, yay, to sign off on it 3 weeks ago after Medicare no longer covered the test. Boo. The PCP still doesn't feel qualified to interpret, but has agreed to let me get the testing. Will I be able to understand the test after listening to this webinar? Actually, it's over, so I don't know. you have to answer that question. I hope so. Now, here's the good news with our lab as opposed to other labs. Any of your doctors, if you've tested with our lab, not with another lab, but with our lab, any of your doctors can call and speak directly to our pharmacist here for free. There's no cost to your doctor, to you, or to your insurance for your doctor after you've been tested to call and speak to a pharmacist. So your doctor could know nothing about this. As long as they're willing to pick up the phone and they have your lab ID number from our lab, they can do that. So it doesn't matter if the doctor has no understanding of it. They can still talk to a pharmacist and figure out how it works. And our results are very easy to understand. And it looks like we have one last question here in 2 minutes, so this is perfect timing. Next question, bupropion, Wellbutrin, worked for me to quit smoking 5+ years ago. Doesn't seem to be working. I guess it doesn't seem to be working now, I gather. Could it be an enzyme interaction or a reaction to another medication I'm taking? I would say if it worked for you in the past, it doesn't work now, it's probably not going to be an enzyme issue. It would probably be, and again, this is just a guess, my first thought would be it could be another medication you're taking that is potentially inhibiting one of the enzymes bupropion is using or inducing it and then affecting it that way. I could be totally wrong. That's just a guess. It's a pragmatic way to look at it as a next step. And you could use those free drug charts online at Indiana University, or you could use our UScript software, which is a pay software, to figure that out. You could look up a drug-drug interaction for that. I think, okay, that's the end of our questions. And John, it sounds like you're on now.
I am on, and I really appreciate all of the valuable information you provided. And wow, I think it was like drinking from a fire hose. there's a wealth of information which is very critical, not only to EDSers, but to many people and most people that are taking various drugs and herbal supplements or vitamins or things like that. So this looks like a fantastic resource that we really need to promote and let people know more about how this all works. So any final words, Dan? We really appreciate you being on the call this evening and giving us this valuable information.
You bet. Well, thanks for the opportunity again. This is pharmacogenetics is extremely important and very much overlooked. And again, it's because it's a new kind of technology. And as patients, I definitely don't want to lead you into the idea that this is the right thing for you and that this is going to fix you or fix all your problems. Keep in mind it can be a helpful thing, and just kind of keep pretty zen about it as you work your way through pharmacogenetics.
Right, exactly. Okay, well, we will have this presentation posted on our site, so you'll be able to see it on our site as well as YouTube. The slides will be there as well. I know many of the slides you might want to zoom in on so that you can get the valuable information that is on there. And I'd like to encourage people to be available next month. We're going to have Dr. Collins available on the 18th to give a presentation. And also, please visit our store, bodysupportstore.com, which funds this program. And we'd like to appreciate those people that have stuck it out with us for the 2 hours, and we're going to call it an evening. So thank you, Dan, and thank you that have attended the webinar this evening. Good night.